Wow, go on, my GCSE science people. Now, in this video, we're going to start looking at biology paper 1, chapter 1, okay? And chapter 1 is referred to as cell structure and support. And this is going to be a revision approach video, okay? For the AQA exam board for GCSE science. Now, this video you can be using it for higher, foundation, or triple, and this is just chapter 1. Now, in this chapter, we're going to be going through the following topics: the world of microscope, animal and plant cells, eukaryotic and prokaryotic cells, specialization in animal cells, specialization in plant cells, diffusion, osmosis, osmosis in plants, and active transport. Okay, so this is going to cover chapter 1. All right, now how should you use this video? Make sure you have your flashcards ready, or you have a book ready to take notes, or you're making your mind map, anything like that. Feel free to pause the video at any point, all right, and make sure that you're commenting and you're subscribing down below. Oh, there's one more topic: exchanging materials. Okay, all right, let's go. So the first thing that we need to know is about the microscope, the different parts of the microscope, and this is called a light microscope, which is the one that is normally found in your school laboratory. Okay, so that is your eyepiece lens, and that normally has a magnification of ×10, and that's the part that you look through. Okay, that's the part that you'll be looking through there. All right, good. Next up, you have your fine focusing wheel, which is right here, and what the fine focusing wheel does is that it moves your stage up and down by small distances. Then you have your coarse focusing wheel, okay? That also moves the stage up and down, but by larger distances, okay? You can see the bigger part there. All right, then you have your objective lens, which is shown right there, and they're normally 3, okay? They normally come in ×4, okay? Times 40 or ×100, okay? Those are the normal magnification. Some may be slightly different. Okay, good. Then you have your slide, which is placed under there, which is what you're going to be looking at. Then you have your stage. Then you have your light source. Okay, good. The only other thing that I would say that you need to know for here is that the magnification that you're looking at an object at is going to be, so the total magnification. Okay, so your total magnification is going to be equal to the eyepiece lens. Okay, so the eyepiece lens. All right, and then you're going to be timesing that by the objective lens. Okay, so for example, what do I mean by that? If you're using the eyepiece lens and you're using your ×4, it's going to be 10 times 4, and your total magnification is ×40. Okay, that would be 400, and that would be 1,000. Okay, good. So that's a different part of the microscope, just for you to have a look there. All right, you should have seen that in the lab. You'd have used that to do your practical. Okay, so next thing that you need to know, there are 2 types of microscope. All right, you have your light microscope, which is shown here. Okay, and you have your electron microscope, which is shown there. Now, your electron, one of the main difference between the electron microscope and the light microscope is that your electron microscope uses a beam of electrons to magnify the specimen that you're looking at. And your light microscope uses light. Okay, now major difference between the two is that your electron microscope, it has a higher resolving power, okay, or higher resolution. Resolution is just the ability to tell the difference between 2 lines. Okay, it also has a higher magnification. Okay, it has a higher magnification, and it is more expensive. Okay, it is more expensive. On the flip side, your light microscope, all right, obviously it's going to have a lower resolution. All right, it's going to have a lower magnification, and it is less expensive. Okay, so it is less expensive. All right, but both of them can be used to magnify different objects. Okay, now the other thing that you need to know now is how to calculate what is called magnification. Now, if you're not really good at rearranging the equation, all right, what I would say is that just remember your formula triangle as shown here, okay? And to remember your formula triangle, this is something that I normally tell the students. I am, okay? What does that mean? Image size is equal to, image size over magnification, and you also have your actual bit there, okay? So this is going to be multiply there as shown there, and that's going to be divide. So once you remember I am, that's all that's you need there. So to find magnification as shown there, magnification is equal to image size, okay, divided by the actual size. Okay, good. So make sure that you know that equation. You must know that equation. Okay, good. All right, now the other thing that you need to do, or need to be able to do, is that you need to be able to do your different conversions. Okay, so you need to be able to convert between millimeter, centimeter, micrometer, nanometer. Okay, now this might seem like a lot of things going on here, but what you just need to remember is that if you're moving from centimeter to millimeter, you're moving from a bigger unit to a smaller unit, you're going to times it by 10, okay? So you're moving from centimeter to millimeter, you're going to be timesing that by 10, okay? If you're moving from millimeter to micrometer, because it's 3 spaces away, you're going to be timesing that by 1,000. And if you're moving from micrometer to nanometer, you're also going to be timesing by 1,000. If you're going the next direction, you're going to be dividing, okay? The next direction, you're going to be dividing by 1,000, okay? That's also going to be dividing by 1,000. All right, good. And you're going to be dividing by 10. All right. These are the common ones that normally comes up on your exam paper. Okay, so those are the common ones that normally comes up on your exam paper. Now, what would a question look like? A question can look something like this. All right. So a scientist observed a cell using an electron microscope. The size of the image was 25 millimeter. You're given the magnification here, calculate the real size. All right, good. So here it is, magnification is equal to image size over real size or actual size. Now you should be able to rearrange this equation. So rearranging, okay, rearranging real size is going to be equal to image size divided by magnification. Okay, and if you're not good at rearranging like that, if your math skills is not good, go back to your formula triangle. Okay, I am, okay, where actual is your real size. So if you need the actual, you cover that. So it's going to be image size divided by magnification. Okay, good. You don't, and please note here, they would like their answer in micrometers. Okay, so they'd like their answer in micrometers. So the first thing that I would do, you could change the millimeters first, or you could do that at the end, whichever one you would like to do. Okay, so magnification, sorry, not magnification. What are you doing? So it's going to be real size, okay, it's going to be equal to the image size, which is 25 millimeters divided by 100,000, okay, divided by 100,000. Okay, but what you need to be able to do, ensure that you are changing your units. Okay, so now we need to change from millimeters to micrometers. So if we go back here, we're moving from millimeters to micrometers. We times it by 1,000. So this needs to be times by 1,000. So that's going to be equal to, okay, you times that by 1,000. So what you're going to have is 25,000 divided by 100,000. Okay, 25,000 divided by 100,000. Okay, and there goes your answer. All right, good. So you're now going to be left with 25 divided by 100, and that's going to give you 0.25. Okay, and you have a look at the mark scheme there. All right, here's another question. Feel free to pause it and have a look here. Now they're asking you to calculate magnification. So remember, to calculate magnification, go back to your formula triangle. Just remember I am. All right, good. So magnification is image size divided by the actual size. Good. Now they've given you the real length, which is 280 micrometers. Okay, which is your actual length. So that's 280 micrometers. You're going to be, and this is now the image size, because that's what you've drawn there. So that's going to be 112 millimeters. But straight away, as you have a look at this, you notice here that you have different units. So you can change the millimeters into micrometers or micrometers into millimeters, any which way that you'd like. I'm going to change my millimeters into micrometers. Okay, so what you can do here, you can remind yourselves. All right, make sure that you have that down here. So you're going from millimeters to micrometers. So that's 1,000. You're going to times it by 1,000. Okay, so you're going from millimeters to micrometers. So it's going to be 112. Just make a note here. We're timesing by 1,000. 112,000 divided by 280. All right, you're going to plug that into your calculator, and that would give you an answer. Give you a magnification of 400. Okay. Normally, when you're writing magnification, you do a multiplication side, so it's going to be times 400. Okay, good. All right, next thing that you need to know, this is a core practical, so make sure that you understand this part of the core practical. You were observing plant cells and animal cells, okay? So make sure that you know the general steps. So when you're doing the plant cell, more than likely you did it with a onion. So first thing that you need to do is to separate the onion into a thin strip. Then you're going to be adding drops of iodine. All right, very important that you realize that. And the iodine is for staining purposes. Okay, you add that onto the onion cell. Then you're going to be lowering something called the cover slip. All right, make sure that there are no air bubbles trapped in between, so that you can be able to see the whatever you're trying to look at. All right, the slide, the specimen. Okay, because that's going to be water. A little bit of water is going to be on it as well. All right. And then you're going to be using filter paper, or you're going to be using any sort of paper just to dry up the excess liquid. All right, now the reason that iodine is being used there, everything is going to look colorless, but remember that iodine basically is going to show up as dye, and it's going to stain the structures so that you're able to see them clearly under the microscope. Okay, now thin, so that light can pass through. Iodine is used so that it is stained. All right, make sure that there are no air bubbles trapped. Okay, now this is what it will look like after you've done put it under the microscope. Okay, and they will normally ask you to do some sort of sketch or drawing. Now when you're doing this, lots of students tend to do that. Okay, but you will lose marks. All right, and I've seen an exam question on this. What you need to be doing is smooth continuous lines just to show up your structure there. All right, you could put your nucleus there. All right, you could do other drawings there. Okay, so it needs to be smooth continuous lines when you're doing your drawing, and make sure that you add some sort of magnification. So are you looking at it at times 40 or times 100, whatever you're doing? Okay, and then when you're labeling, just nice straight lines when you're labeling. Obviously that's going to be your cell wall, because we're looking at a plant cell. On the inside, don't know if you can see that clearly, you would have your cell membrane as well. Okay, so just remember that some of the rules that when you're doing your drawing. Okay, so the rules include magnification, do not shade, and draw the correct cell shapes. Do not include gaps between cells. Okay, all right. Now, when you're doing animal cells, we normally do cheek cells. Okay, good. And you use a cotton bud to scrape the inside here. You're going to wipe that on your glass slide. And this time, we're not going to be using iodine. We use methylene blue or methyl blue. Okay, and again, that's a dye which is used to stain the structures. Otherwise, the structures are going to look colorless. Okay, good. All right. So this is what that would look like. Those are your cheek cells. So clearly you can see the nucleus there, quite obvious to see, and you can see the cell membrane. Okay, excellent. All right. Now you need to know the different steps. I've seen 6 markers on this, the different steps for using the microscope. Okay, you always start with the smallest objective lens, the one with the lowest magnification. All right, then you turn your coarse focusing wheel to move it up and down to make sure that you have as small as possible gap between the stage and what you're trying to look at. When you're finished with that, your slide is going to be placed underneath this, on the stage, underneath the objective lens here. Okay, and then you adjust the light source to make sure that you're seeing it correctly. Then you're going to be looking through your eyepiece lens. Okay, focusing the coarse wheel until you see it. All right, you want to ensure that it is clear and sharp. All right, to see the image bigger, you're going to change your objective lens. Okay, so you're going to move from your lowest, all right, to a higher objective lens, and then you're going to be using your fine focusing wheel. Okay, these are some good steps that you need to know. Make sure that you know the steps here. You can pause the video, make notes, okay, as you go along. All right, next up, now we're looking at plant cells and animal cells. All right, make sure that you know how to at least do a sketch and you know how to label the different parts. Okay, so please note that there are some structures which the plant cell have that the animal cell does not. Okay, one structure is the cell wall, another one is the vacuole, the other one is the chloroplast. Okay, make sure that also you know the functions of these different parts. All right, we're going to be looking at that next. Okay, so to do the function, I would suggest that you probably draw a table looking something like this. All right, and then after you've drawn the table, you're going to use the next couple of slides just to populate that table. Okay, so you have your nucleus, your cytoplasm, cell membrane, mitochondria, ribosome, cell wall, chloroplast, vacuole. Okay, so first up, let's look at the nucleus. So the nucleus, all right, that controls the activity of the cell. It contains the gene and the chromosomes, okay, and it is needed to make new cells and new organism, and this is found both in the plant cell and the animal cell. All right, next up, you have your cytoplasm. This is where most of the chemical reaction takes place in the cell, okay, and it is found both in the plant cell and the animal cell. Cell membrane, it controls what comes in and out of the cell. Okay, good. This normally happens by diffusion, osmosis, or active transport. We're going to be looking at that later. Okay, good. We're still with the animal cells, also found in plant cells as well. So you then have your the mitochondria. The mitochondria is where respiration takes place. Okay, where that means that oxygen and glucose is reacted here to release energy. Okay, so this is your site of aerobic respiration. All right, next up, ribosomes. This is where protein synthesis takes place. All right, so all the protein is made there. That is found both in animal cell and plant cells. Okay, however, you have some structures that was mentioned earlier, which is found in the animal cell but not in the plant cell. These structures include the cell wall, okay, made of cellulose, and it gives the cell structure and strength and support. Okay, good. And this is found in the plant cell only. Okay, good. Chloroplast, this is the site of photosynthesis. It also contains some green pigment which is called chlorophyll. Okay, so we could add that as well. It contains green pigment, which contains chlorophyll, and that is responsible for trapping of light. Okay, so trapping of light. You could add that to your table as well. Then you have your permanent vacuole, which is filled with cell sap. All right, and that is important to support the plant by keeping it rigid. Okay, good. So make sure that you've used that to fill your table in. Okay. All right. So the other thing that we need to be looking at now is prokaryotic and eukaryotic cells. Okay, make sure that you know the difference between the two. So prokaryotic cells, these are cells such as bacteria. All right. Viruses are also prokaryotic, but the important thing that you need to know is that they do not have membrane-bound organelles. The word organelles just means the structure. Okay. So nucleus is an organelle, mitochondria is an organelle. Those are organelles. So they do not have any membrane bound organelles. Okay, that means that they do not have a distinct nucleus. They don't have mitochondria, chloroplast, or anything like that. They're not found in an organelle. Okay, good. So that's prokaryotic cells. Whereas with the eukaryotic cells, okay, so for your eukaryotic cells, those would have membrane-bound organelles. Okay, so those would have membrane-bound organelles. Okay, which means that they have nucleus, so their DNA is found inside a nucleus. So DNA found inside nucleus. Okay, DNA found here. Their genetic material is found there. Also, prokaryotic cells would be older and smaller, whereas eukaryotic cells would be newer and they would be also larger. Okay, so here is a diagram of a common bacterial cell that you need to be able to describe. Okay, so if you notice here, the genetic material, there is no nucleus. So normally it is found in a nucleus in the eukaryotic cell, but there is no nucleus found here. All right, notice that you also have some extra loops of DNA. Need to be familiar with that. That's called a plasmid. Okay, then you have your ribosomes. You remember that's for making protein. You have your flagella for movement. You have your cell membrane, and you have your cell wall. Okay, so bacteria, they have plasmids or loops of DNA which are not contained in a nucleus. Okay, they can also produce clones of themselves. All right, so here's another look, a more 3D look of the bacterial cell. You can see the different functions and different information about the bits there. So again, chromosome not found in the DNA. That's the most important thing that you need to know about the eukaryotic cell. Sorry, prokaryotic cell. My bad. Prokaryotic. So remember, your eukaryotic now, they would have nucleus. Okay, so the eukaryotic cells, they would have nucleus. All right, so now we're going to be looking at specialized cells. Okay, now what do we mean by specialized cells? Specialized just mean that they have different structures for carrying out a particular function. Okay, so multicellular organisms, they have different structures, and those structures are able to carry out different function. So in order to do this, they need to have special features, special adaptation, meaning that these cells, they would look quite different. Okay, good. And the process in which cells become specialized, that's called differentiation. So I mean, this is going to come up in chapter 2, but just to be clear, all the cells started with unspecialized cells. Okay, and there's a special name for these unspecialized cells. All right, and these unspecialized cells, they can differentiate into different cells. So it could become a sperm cell, it could become an egg cell, it could become any sort of cell, red blood cell, any sort of cells. Okay. All right, so it's very, very important that you know that these unspecialized cells, that process, they differentiate, okay, into becoming your specialized cells. All right, and now we're going to be looking at your different specialized cells. So the first one that we're going to be looking at is the nerve cell, the neuron, and the function of this cell is that it carries electrical impulses around the body. Okay, now adaptation, what makes them so good at their job? They have lots of connection to other cells. So you can see the dendrites here, they can be connected to several other types of cells. They have a very long axon right here, which is shown there, all right, which can run and carry information long distances. Also, it has lots of mitochondria, which provides energy, and also there's something called the myelin sheath. Okay, the myelin sheath, what it does, it acts as an insulator. Okay, that's what makes it good at its job. All right, good. Next up, muscle cell brings up movement. All right, in terms of adaptation, they have special proteins that can slide over each other, lots of mitochondria, which obviously is going to provide energy through respiration, and it also has a store, a glycogen store, which can be quickly broken down to release glucose, which is needed for respiration. Okay, then we have our sperm cell. The function there is to carry genetic information from the male part of the plant. Sorry, I almost said plant, from the male parent during reproduction. All right. So adaptations. The adaptations now would include the following. It has a long tail for swimming. Lots of mitochondria is packed inside the head, which transfers the energy which is needed for swimming. It also contains digestive enzymes right at the front of the head there, which is used to break down the layers of the egg. All right, and it also has a large nucleus which contains the genetic information. All right, it would have the chromosomes inside. All right, now all of those that we were looking at, those were animal cell. Now we're looking at the root hair cell. All right, so the root hair cell, its function is to absorb water and mineral ions. Okay, water and mineral ions, which is then going to be transported up the plant. All right, so adaptations, it has a large surface area, and it asks why there, and the reason that it has a large surface area is so that it can absorb as much water and mineral ions as possible. All right, next up, it has a large permanent vacuole. So look at the size of that vacuole there, which speeds up the movement of water by osmosis. It also contains lots and lots of mitochondria, and the reason for that is sometimes it needs to transport mineral ions against its concentration gradient. We're going to be looking at that later, but energy is needed for this, for active transport. Okay, good. Next up, you have your photosynthetic cell. All right, normally referred to as your palisade cell. All right, now these cells contain lots of chloroplasts. Why would it contain lots of chloroplasts? For photosynthesis, to trap light. Okay, for photosynthesis. Remember, these are found inside plants. It also has a large permanent vacuole, okay, which keeps the cell rigid as a result of osmosis. Good. And it also has a regular shape, all right, so that it allows the light just to pass down there. Next up, you also now have something which is called your xylem cell. All right, specialized cell. Your xylem cell is responsible for carrying water and mineral ions, all right, from the root to everywhere in the plant. Okay, it's normally found with lignin, all right, and these are dead and they're hollow, all right, and water is transported through that via capillary action. Then you have your phloem cells. Now your phloem cells is responsible for carrying the sugar which is made by photosynthesis, and that sugar is glucose. Okay, good. And the adaptations include the fact that the cell walls are quite thick. Okay, good. So the cell walls between the cells break down to form something called the sieve plates. Okay, so these are your sieve plates here. All right, good. And the food is moved in both directions. Okay, it also contains lots of internal structure which is supported by a companion cell. So this right here is your companion cell, okay, which keeps it alive. All right, and also contains mitochondria in the companion cells. So there will be mitochondria there, which helps for the movement of food up and down the phloem cell. Okay, so just remember, phloem transports the food and the xylem transport the water and mineral ions. Okay, good. So those are like your specialized plant cell and specialized animal cells there. All right, next up now we're going to be looking at the topic of diffusion. Now diffusion, most of you already know this, but pop that down. So diffusion is the spreading out of gas particles, all right, in a substance or in a solution, all right, and it's moving from a region of high concentration to a region of low concentration. So right here you have high, then on this region you would have low. All right, so you're moving from high to low, okay, and no energy is required. So no energy needed, and this means that it's a passive process, okay? So that is diffusion. Now, can you think of some factors which would affect diffusion? So there are some little clues there that should help you. So first factor that we're going to be looking at is concentration gradient. Now, look at these two diagrams. You have diagram A, you have diagram B. All right. Now, which one do you think would have diffusion happening at a faster rate? Do you think it's going to happen faster in B or A? All right. You should be saying A. And the reason that diffusion will happen faster in A, look at the number of blue particles that you have here. 1, 2, 3, 4, 5, 6, 7. And I have none on this side. So the difference in concentration is quite high. And that difference is what we refer to as concentration gradient. So the concentration gradient, in your textbook, they would refer to it as being steep. All right. So there's a large difference, and the blue particles are going to diffuse across there. All right. Now, if you look here, 1, 2, 3, 4, 5, 6, 7. 1, 2, 3, 4. The concentration gradient is not very steep. So yes, you will have the blue particles going across, but it's going to happen slower, okay? And that is concentration gradient, okay? So the bigger the concentration gradient, the faster the rate of diffusion, all right? Good. That's it there. So concentration gradient is a major factor. It would diffuse faster from a very high concentration to a low concentration gradient. And the steeper the concentration, the quicker the rate of diffusion. Next up, temperature. That should be fairly straightforward, okay? The higher the temperature, the more energy the particles have, and that energy is what we refer to as kinetic energy. So they're going to have more kinetic energy, which means that they're going to be moving faster. So that increase in random movement, which means that the rate of diffusion is going to be quicker, okay? And that's temperature. And then surface area, all right? The greater the surface area, the faster the rate of diffusion. So if you notice here, there are more points of contact here, okay? Whereas if it's flat, you're going to have less points of contact. So there's less surface for diffusing, okay? Good. Next up, we're going to be looking at osmosis. Now, osmosis is the diffusion of water molecules, okay? You must mention this word in your definition. It's a diffusion of water molecules, and you're moving from where you have a dilute solution, so you have lots of water, lots of H2O, to a concentrated solution where you have very little H2O, and it must be happening through a partially permeable membrane. So those are some key bits that you must have, okay? You must ensure that you have water, and you must ensure that you have partially permeable membrane in your definition, okay? Good. So these are my water molecules, which is shown there. Okay, let's get our pen back. So these would be my water molecules. All right, so you're moving from a dilute solution to a concentrated solution. Okay, so you're moving from a dilute solution in and out of the cell. All right, so there are some key terms that you need to know. Okay, so the first term that you need to know is isotonic. Okay, so you need to ensure that you know these key terms here. So the term isotonic means that the concentration on both sides are the same. So if you look at this side and you look at this side here, my concentration of salts are the same. So iso, the prefix iso means same. Tonic means salt. Okay, so if you notice here, we have 1, 2, 3, 4, 5, 1, 2, 3, 4, 5. The concentration on both sides are the same. Okay, which means that the net movement of water is going to be equal. Okay, so the net movement of water in isotonic solution is going to be equal. All right, now let's look at another term, hypertonic. So the prefix hyper means high, okay, and tonic refers to salts or solute. Okay, so it refers to salts or solute. Okay, good. Now if you have a look here, on the outside of the cell, I have lots of solute particles, okay? And on the inside of the cell, I have very little solid particles. 1, 2, 3, okay? Now, where does water move? Remember, water always moves, right, from where you have lots of water molecule to where you have a little bit of water molecule. So the net movement of water will be outside the cell, okay? So water is going to be moving out of the cell like that. Okay, good. And that's called a hypertonic solution. And then you're now going to have something called hypotonic. So hypotonic means that the prefix hypo means that you have low. Tonic again means solute or salt. Okay, so you're going to have low solute, okay, in relation to each other. So in this scenario, the water particles are going to be moving in that direction. They're going to be moving into the cell, okay, good. Now here are three different diagrams with hypertonic, isotonic, and hypotonic, right? So if you notice here in their hypertonic solution, notice that the cells, they become shriveled, okay. All right, and right here, if you notice here that one of your cells and your hypotonic, it becomes burst. Okay, so it releases water. Okay, so it bursts, and the reason for these cells to burst is because you have lots of water going inside. It becomes bigger. It has no cell wall, so it's going to burst. All right, because the water is going to move in. Isotonic, you have the same amount of water moving in and out, so the cells remain same size. So the cells here would remain the same size. Okay, these cells would become bigger and perhaps burst only if it's animal cell. All right, plant cells would not burst because they have a cell wall, so these become bigger. They would burst. All right, but that is only animal cell. Only the animal cell would burst. All right, plant cells would not burst, and these one now become shriveled, or in some textbook you see they use the term flaccid. Okay, becomes flaccid, and these become turgid or tough. All right, good, good. And then you see this term as well, plasmolysis, okay, which just means, and that term would be used only for the plant cells. So plasmolysis just means that the cell membrane becomes pulled away from the cell wall. Okay, so make sure that you know these different terms: hypertonic, isotonic, and hypotonic. Okay, good. Now, all right, what should we look at here? Just as something for you to practice. I'm not going to go through everyone, but just think about where is the water going to be moving. So what I have here, I have a cell. Okay, good. And then I have my beaker here. That would be my beaker. Look at the difference in concentration here. So if you notice here, 90% water, 85% water. Water always move from where you have a high amount of water to low amount of water. Okay, good. So the water is going to be moving out. And what would we describe this solution as? So we have a higher amount of solute on the outside. So I would describe this solution as hyper. Okay, so feel free to go ahead and try the rest of these. You can pause the video, okay, and then the answers are all there. Okay, so looking at whether it's hyper or hypo. If you have a look at the second one here, all right, you have more water on the outside, less water on the inside. The water is going to move in. You have less solute on the outside compared to inside. So therefore, it is going to be hypotonic. Okay, look at this one. That one is isotonic. Okay, good. Now, another thing that is commonly asked from this chapter here is calculating percentage change. Okay, and this is leading up to the potato practical. All right. Now, to calculate percentage change, okay, to calculate percentage change, it is going to be the change in mass, okay, over the initial mass, and you're going to be timesing that by 100, not 1,000 as I wrote there, but it's going to be times by 100. Okay, so what do you have here now? Your change in mass. So your dry potato was 21 and the potato in the water was 27. So let's look at this one here first. Okay, so what you're going to have here, you're going to do 27 minus 21 divided by whatever your initial mass was. So your initial mass was 21. You're going to times that by 100. So that's going to be 6 divided by 21, and you're going to times that by 100. And let's do that quickly on our calculators. All right, so that's going to be times by 100, and that's going to be 28.57%. That's the percentage change in mass. So it's increase in mass by 28.57%. Now look at what happened when the potato was placed inside the salt water. The mass decreased. Okay, so let's look at that one. So it was 21 at first. We'll just clear this so that you're not good. So this time we're now looking at this change here. So it was 21 at first and it's now at 19. Okay, so it's going to be 19 minus 21 over 21. So yes, I know you're thinking it's going to be a negative number, which it is, because initially it was 21. The final mass is 19. So that is a change of -3. Okay. Sorry. What is GCSE boss saying? All right. It's going to be a change of -2 over 21, and you're going to times that by 100. Okay. So -2 divided by 21 times that by 100, and that is a minus 9.52 percentage change. Okay. They love asking this question, especially if it comes up for the osmosis practical. Okay, good. So we've already calculated that one there. All right, so we got minus 9.52. That is done. All right, that means that the potato has lost a mass. Now, why are we talking about percentage change? Now, you would have done this practical here, osmosis in potato strips. Okay, so what's happening here is that you're given potatoes of not necessarily the same mass. Okay, so you're given some potato there. They're placed in different solution. It can be sucrose, it can be salt, it can be some type of sugar. Okay, and notice now that we are using the same volume of solution. So you got your potatoes, you measure the mass initially, you place them into your different solution, and you leave it for a couple of minutes. By right, you should be covering it. Okay, and the reason that you need to cover it because you don't want it to evaporate. All right, because if it evaporates, it's going to change the concentration, which can give you incorrect reading. Okay, you leave it for some amount of time. All right, and then you take it out, you blot dry on paper towel. Very important that you know that you blot dry on paper towel. All right, and then after you blot dry, all right, you're going to take the mass after. Okay, and when you take the mass after, you can then work out the percentage change in mass like what we were doing before. Okay, now what's going to happen is that in some of these, water is going to move into the potato, or some of it water is going to be moving out of the potato through that semi-permeable or partially permeable membrane. Okay, so When you're finished, you would have results looking like this, okay? So you will have your different solution, you have your starting masses. So if you notice here, your masses aren't the same, and that's the reason why we need to calculate the percentage change in mass. So here is one that you could also try for yourself. So how would you work out the percentage change in mass? We've just done that before, okay? So it's the change in mass here, 0.15. divided by whatever you started out as, 1.35. You're going to be timesing that by 100. So it's going to be 0.15 divided by 1.35. Good. You're going to times that by 100, and you should get roughly 11.1%, and that is recurring. Okay. Good. So if you notice, some of these have a negative change in mass, some of these have a positive change in mass. Those that has a negative change in mass, those solutions would have been hypertonic. So the water is going to be moving out of the potato. Okay, good, and into the different solution. That's why the mass decrease. These here, that has a positive one, those would be hypotonic. Okay, good. Now, after this is done, right, you need to think about what your variables are as well when you do this practical. So your independent variable is whatever you're changing. Okay, so just remember, independent variable is whatever you're changing. So what are we changing in this experiment? We are changing the concentration. You're changing the concentration of solution. Okay, your dependent variable is what you measure. So what would you be measuring? You're measuring the change in mass of the potato. Okay, so the change in mass of potato. Okay, and finally, your controlled variable is whatever you're keeping the same. So we're keeping the volume of solution the same. Okay, volume of solution, you're keeping that the same. Are we keeping anything else the same? Yeah, the time that the potato is spent in the solution, we're also keeping that the same. Okay, but those are your variables there. All right, now we're now going to be looking at, so we've looked at diffusion, osmosis. Now we're going to be looking at active transport. Now in active transport, have a look at what's happening here. Look at these orange molecules. I have 4, okay, on the outside, and I have 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 orange molecules on the inside. Okay, so if you notice here, your concentration gradient is 9 to 4 here. Okay, so this has a higher concentration gradient, and this one has a lower concentration gradient. So this one is higher, that one is lower. But look at where the particles are moving. The particles are moving from low to high. And that is what we refer to as active transport. So active transport is the movement of particles from low concentration to high concentration, and energy is needed. So it is an active process. Okay, so energy is needed, and that energy is normally provided in the form of something called ATP. Okay, so active transport is like riding a bicycle up a hill. You're going against the concentration gradient. So if you look, the concentration is going down here, but you're going against it. You're going up, which is why energy is needed. And you also use special carrier molecules there. Okay, now there are lots of examples where active transport takes place, and you need to know that. Okay, so glucose is moved from the intestine into the blood, and this takes place in the villi or villus. Okay, that is done by active transport. Inside root hair cells, which we spoke about earlier, that is done by active transport. So mineral ions moving from the soil into the root. All right, good. And then there's some animals as well. Crocodiles, for example, they have special salt glands which can move substances by active transport. Okay, so make sure you know the difference now between active transport, osmosis, and diffusion. Okay, so osmosis involves water only, requires energy, that is active transport. Passive is both for osmosis and diffusion. All right, this is very nice for your flashcard. Okay, movement of particles. All of these involve the movement of particles. Need a partially permeable membrane or semi-permeable membrane. That is both osmosis and active transport. You're moving from high to low concentration, so you're moving down the concentration gradient. That is diffusion and osmosis. Against the concentration gradient, that is active transport. All of these occurs in nature. Mineral ions moving into root hair cell, that is active transport. Leaves, clearly, that is diffusion. Anything to do with water, that is going to be osmosis. Movement of solutes, diffusion, and active transport. Okay, good. Now, the final bit that we're now going to be looking at, this is going to be now the final part for chapter 1, which is exchanging materials. Now, you need to be able to work out something called surface area to volume ratio. Okay, you need to be able to work on something called surface area to volume ratio. Very important that you're able to do that. Now, to work out the surface area, it is the area of one side. All right, but because we have 6 sides here, so to work out the area of one side is going to be 1 times 1 times by 6. Okay, so it's going to be 1 times 1 times 6, so that's going to be equal to 6 there, and that's going to be 6 centimeter square. Okay, then to work out the volume is length times width times height, so it's going to be 1 times 1 times 1. Okay, good, and that's going to give you an answer of 1 cm cubed. So what's my surface area to volume ratio here? It's going to be 6 to 1. Okay, let's look at another example. All right, so now we're now increase this side, this cube here to 2. Okay, so all the sides are now 2 centimeters. Okay, so now it's going to be 2 times 2 times by 6. Why are we times it by 6? Because you have 6 different sides. Okay, it's going to be 2 times 2 times 6. That's going to give you an answer of 24 cm squared. Good. Then to work out the volume, it's going to be 2 cubed. So it's going to be 2 cubed, which is going to be 8. So what's my surface area to volume ratio? It's going to be 24 to 8. But of course, we would like to get this number, your volume, to be in 1. Okay, so we'd have to divide through by 8, and therefore it's going to be 3 to 1. So do you notice something that's happening? You can try that one on your own. Okay, however, you should realize that the bigger object becomes, the smaller its surface area to volume ratio. That's a key point. Okay, please make a note of that. The bigger the object, you can see that, you can check your answer there. Bigger the object, the smaller the surface area to volume ratio. Okay, the smaller the surface area to volume ratio. Now, why is that important? Okay, this is very important because when you think about exchange surfaces, if you think about something like a single cellular organism or a unicellular organism, they have a high surface area to volume ratio, so they can absorb substances quite quickly and quite easily. But when you think about multicellular organisms such as ourselves, they have a lower surface area to volume ratio. Therefore, it is harder for these organisms to absorb substances. Okay, so that means that they require specialized exchange surfaces for transport. Okay, these don't require specialized surfaces. This can happen by osmosis, by diffusion, or even active transport. Okay, diffusion, not diffusious, diffusion or active transport. Okay, good. Whereas with multicellular organism, we would need special circulatory system. Okay, so common questions that they like to ask about this for exchange surfaces. what makes it suitable for its job? And here are some common mark scheme answers. You would say that they're thin, so that the diffusion can happen quite quickly. They have a large surface area, so that having that large surface area would allow maximum amount of diffusion or exchange to happen. It would have lots of blood vessels to maintain a high concentration gradient, okay? Maintain a high concentration gradient, and also it would be well ventilated, okay? They have good ventilation. So, common question that they would ask: why is the alveoli suitable for its job? First thing that you need to be saying is that it has a large surface area, all right, which means that it can allow for the rapid movement or exchange of gases. It also have thin walls so that it happens quickly, okay? And it also has a moist lining to allow the gas to dissolve. So those are your key points that you need to know for that exchange surface there. Another common example, the villi, okay? This is where diffusion is occurring, okay? And, well, or sorry, substances are absorbed in digestion. That's the word I'm looking for. Brain freeze, okay? So what's happening here? Why is it suitable? Again, surface area is quite high, very thin walls, only one cell thick, and they have a very good blood supply, okay? They have a very, very good blood supply, which maintains their concentration gradient, okay? So this is the end of chapter 1, okay? Make sure you go on to chapter 2. That video is going to be on my channel. Make sure that you like, share, and subscribe. I'm out.